So hello everyone, welcome back to Hasvora Education. Today we will be solving the biology paper 3 of the May June 2025, and this is the 3-4 variant. The time allowed for this paper is 2 hours. So what I'll be doing in this paper is that I'll be just moving on with some random values, okay, so that we can actually perform the calculation, and we'll be also looking over the whole paper, okay, so how to attempt the question, how to make the paper, the tables, okay, how to arrange the data, and how you are going to be reducing the conclusions according to the question, okay. So we'll be not performing any experiment. So before I start the video, I would like if you can just subscribe the channel and just like the video and share with anyone you think it will benefit, okay. So let's move on to the first question. So question number 1 says that catalase is an enzyme that is found in the yeast cells, okay. So most of you will already know this, that catalase is in the yeast cells, and catalase catalyzes the breakdown of hydrogen peroxide to produce water and oxygen, okay. So those who study chemistry already know this, because they have on some part of the paper they have actually seen that hydrogen peroxide is actually broken down into the water and oxygen, okay. So now they are telling you that you will investigate the effect of copper sulfate on the progress of the reaction, okay. So now why are we actually investigating the effect of copper sulfate, okay. So what is copper sulfate doing here? So copper sulfate can either be, now there are two types of possibilities when you are doing this type of question, okay. So it can either be the inhibitor, okay, it will stop, or you can say it will decrease the activity of the enzyme catalase, or it can also act as a coenzyme, okay, and in that case it will actually you can say it will accelerate the reaction, or the reaction will not be possible without it, okay. So you have to investigate the progress of this reaction, and now they're telling you that you will do this by stopping the reaction after 5 minutes. So you are actually checking the activity for 5 minutes and measuring the concentration of hydrogen peroxide, okay. So now here the examiner is a little bit trickier. Why? Because he is checking the reactant, okay, that how much the reactant is converted into the products, okay. So in the case, so if there is low hydrogen peroxide concentration, so what will actually be reduced? We will reduce that there has been much activity of the catalase, and most of the hydrogen peroxide has been converted into water and oxygen. But if there is a high hydrogen peroxide concentration, this means that the catalase activity has been reduced, and the reactant has not been converted, okay. Because most of the time he actually checks the activity by the products, okay, but here he's a little bit trying to confuse you with that, okay. So now here I know that because I will not be doing the experiment, and because you will be investigating the effect by doing the experiment, so I know that it is the inhibitor, okay. Copper sulfate is the inhibitor of catalase. So what does this mean? Okay, so this means that if I increase the concentration of the copper sulfate, so what will happen? The hydrogen peroxide concentration, which is H2O2, its concentration will also increase. Why? Because it is inhibiting the catalase, and that will reduce the activity of the enzyme, and in that case what will happen? The reactant will not be converted into the products, okay. So I just told you because I will not be doing the experiment, okay. Now they are telling you that potassium permanganate is used to measure the concentration of the hydrogen peroxide. Let's see how we are going to be measuring it with the concentration of hydrogen peroxide with potassium permanganate. You are provided with the material shown in the table 1.1. So they have given you the yeast suspension, and it will actually have the catalase enzyme, okay. You have hydrogen peroxide solution, you have 1% copper sulfate solution. So now when we are actually trying the you can say the effect of different concentration of copper sulfate, the effect of it on the catalase enzyme. Now what we will be doing, possibly the examiner will now tell us to actually either do the simple dilution or the seal dilution with the copper sulfate, okay. So Now then we have the dilute sulfuric acid, we have the potassium permanganate solution to measure the concentration of H2O2, the hydrogen peroxide. Okay, so if any solution comes in contact with your skin, wash off immediately under cold water, and you should wear suitable eye protection. It is recommended that you wear gloves when using A and P. Okay, so let's see now. Now you will need to carry out a serial dilution. Okay, so now he has specified that you have to carry out what? You have to carry out serial dilution of 1% copper sulfate solution C to reduce the concentration by half. Okay, so now sometimes he will tell you to do the serial dilution, but he will tell you to reduce the concentration 10 times. Okay, so in that case you have to then use the formula. Now when we are actually reducing the concentration by half, what does this means? This means that we have to add same volume of copper sulfate and water each time. Okay, so for example, if I have 1%, so now if I'm going to 0.5%, what I will do, so in the case if I'm preparing 20 cm cube of 0.5%, what I will do, I will just take out 10 cm cube of 1% copper sulfate solution, and then I will add 10 cm cube of water to it, so it will become 0.5%. Okay, and in the case if most of you are actually confused with this fact that how it becomes half, you can just use this formula. Okay, this formula is M1 V1 is equals to M2 V2. Here the M1 is actually the concentration that you are using. This is the concentration you are using. Okay, then this volume is the volume you use. Okay, this is the concentration you want to prepare. Okay, and this is the volume that you want to prepare. Okay, so in the case now if we want, where the concentration we are using R is 1%. Okay, now we do not know what volume we will pick out. Okay, so let's just keep it as V1, and now we want to prepare the concentration of 0.5%. okay, which is the half of it, and now we want to prepare 20 cm cube, or you can say 20 cm cube of 0.5 concentration, it becomes 20. Okay, so what will V1 be? V1 will be 10 cm cube. Okay, so it will be 10 cm cube. So now most of the students are just confused with this. Okay, they think that you have to pick out 20 cm cube of the 0.5%. Now 20 cm cube is the final volume. Okay, it's the final volume that you have to prepare. So if you are actually using 10 cm cube of the 1%, this means that you have to add 10 cm cube of the water to it. Okay, so I hope you understand this. But just remember that in the case if you are doing serial dilution and you are reducing the concentration by half, you have to add 10 cm cube. In the case if you are preparing 20, so you have to add 10 cm cube of that concentration and 10 cm cube of water. Okay, so now they are telling you that after the you will need to prepare, so you will need to prepare 4 concentration of copper sulfate in addition to 1%. Okay, so in addition to 1% we have to prepare 4 concentration. This means that we have total 5 concentration. So if I have 1% as the initial, I have to prepare 0.5%. okay, just divided by 2, then divide 0.5 by 2, it becomes 0.25%. okay, then again 0.125%. so now we have 3 additional concentration, so one more, then we will have 0.0625%. okay, so these are the concentrations that you have to prepare. Okay, now they are telling you that after the serial dilution is completed, you will need to have 10 cm cube of each concentration available. Now if you see, they have given you 20 they have given you how much? They have given you 30 cm cube. So out of that 30 cm cube, what you will do, you will actually use 20 cm cube which will be the initial 1%. Okay, this will be the initial 1%. So you will have 20%, 20 cm cube of 1%. This will be 20 cm cube of 1%. Okay, and then you will actually be transferring 10 cm cube. Okay, and then you will be adding 10 cm cube of H2O, your water. So if you transfer 10 cm cube of 1%, so how much will be remaining? You will be remained with 10 cm cube of 1%. And now when you add 10 cm cube of 1% and 10 cm cube of water, you will be having 20 cm cube of 0.5%. Okay, and then you will be doing the same the thing you did with the 1%. Okay, you will be transferring 10 cm cube and you will be adding. So in the end we will have 10 cm cube of each of the concentration. Okay, hope you understand this. So we will need to have 10 cm cube of each concentration available to use. Now they are telling that complete figure 1.2 to show how you will prepare your sealed dilution. Each beaker should have a labeled arrow to show the volume of copper sulfate solution transferred, a labeled arrow to show the volume of distilled water W added, and a label under each beaker to show the concentration of the copper sulfate solution. Okay, now these are 3 easy marks. Okay, these are for the drawing and you should gain full marks in this. Okay, so now just follow the procedure. So now you have 20 cm cube of 1% copper sulfate solution. Now this is the original. Okay, this is the original that have been given to you. Now the arrow that you are seeing, this arrow, this is used for the transfer, the thing that you are transferring. So what are we transferring? We are transferring 10 cm cube of 1% copper sulfate solution. Okay, I am just writing the formula, but you do not have to write the formula, you have to write it full. Okay, let's just write it fully. Okay, so 10 cm cube of copper sulfate solution. Okay, I am transferring 10 cm cube of copper sulfate solution, and then what I will do, I will also add 10 cm cube of water to it. Okay, this is for the water. I am adding 10 cm cube of distilled water W. Okay, and the copper sulfate solution was C. So what will I will be obtained with? I will have 0.5% of copper sulfate solution. Okay, this will be the thing I will be actually having at the end of this. Now I will do the same procedure with the 0.5%. Okay, what I will do is I will again draw an arrow like this. Okay, that is being transferred, and let me just adjust it a little bit. So it will be like this. Okay, and this will be 10 cm cube of 0.5% copper sulfate solution C. Okay, and I will be adding 10 cm cube of water to it. cm cube of distilled water, which is W, and what will I will be obtained with? The concentration will become half, so this will become 0.25% of copper sulfate solution. Okay, so 2 more time, let's just do it again. Okay, so like this, and then we have to transfer it. Okay, so this will be 10 cm cube of 0.25% of copper sulfate solution C, and this will be your 10 cm cube of distilled water W. Okay, and what will I will be obtained with? This will be 0.125% of copper sulfate solution, and then once again I will transfer this, and I will have, let's just adjust this a little bit. Okay, so this will be something about this, and this time I am transferring 10 cm cube of 0.0625% of copper sulfate solution C. Okay, and this will be 10 cm cube of distilled water W, and at last I will be obtained with 0.0625% of copper sulfate solution. Okay, so this will be the whole diagram. Okay, you just have to make it like this to gain full marks. Okay, so now you must see that initially I have 20 cm cube of it. Okay, so if I transfer 10 cm cube of it into the next beaker, it will be left 10 cm cube, and same for the other ones except for the last one. Okay, this will be 20 cm cube only. Okay, because from this you are no more transferring. Okay, so but it is not a problem. Okay, don't need to worry about that. Now let's just read out the steps. So carry out step 1 to 7. So prepare the concentration of copper sulfate solution. So we have already prepared that. Okay, let's just assume. Then step 2 says that label test tubes with the concentrations prepared in step 1. Okay, so now you will be given the test tubes in your lab. Okay, so if for example if you have 5 to 6 test tubes, you will label them like this. Okay, so if you have a test tube like this, you will just label it with a marker as 1%, then the other one with 0.5%, and so on like that. Okay, with 0.25, 0.125, 0.0625%. Okay, and now they are telling you that put 1 cm cube of 1% copper sulfate solution into the appropriately labeled test tube. So what you have to do, you have to put 1 cm cube of 1%, you have to put 1% of the copper sulfate solution into the test tube that has been labeled 1%. Okay, so you have to only add it to the test tube that has the 1%. Okay, now the examiner is only telling you how to do the thing. Okay, so then put 1 cm cube of each of the other concentration of copper sulfate solution as prepared in step 1 into the appropriately labeled test tube. Okay, so now you have to follow the same procedure. You have to put 1 cm cube of 0.5% into a test tube of 0.5%, the 0.25% into its respective test tube. Okay, and same for the other ones as well. Okay, now what you have to do next, you have to stir the yeast suspension. Okay, so actually it mixes well and the temperature is constant for the catalase enzyme in the yeast, and then you have to put 1 cm cube of the yeast suspension Y into each of the test tube. So now you have to put this suspension into the each of the test tube. Okay, because this is the enzyme, okay, for which you are measuring the activity. And shake the test tubes gently to mix. So now you have the you have the copper sulfate different concentrations and the yeast suspension of 1 cm cube in each test tube. Okay, now what you have to do, you have to put 1 cm cube of the hydrogen peroxide solution, which is actually your substrate or the reactant in this reaction, into each test tube and shake it gently to mix, and then wait for 5 minutes. Okay, so now what we are doing, we are actually allowing constant time for activity of enzyme, and it is the activity with different concentration of copper sulfate. Okay, now after 5 minutes you have to put 2 cm cube of sulfuric acid into each test tube. Okay, so now why are you adding 2 cm cube of sulfuric acid? So you are doing it just because to stop the reaction, or you can say the hydrolysis of the hydrogen peroxide to water and oxygen. Okay, because you were meant to measure the activity for 5 minutes only, so after that you have to stop it. Okay, so now the examiner is also telling you this, that the addition of sulfuric acid stops the breakdown of hydrogen peroxide. Now you will compare the concentration of hydrogen peroxide remaining in each test tube using potassium magnate solution. Okay, so now till now, how much of the hydrogen peroxide that has been hydrolyzed, you just have to check the concentration of that. Okay, you just have to check the concentration of that. And just because now we know that the copper sulfate is the inhibitor, so what will happen, the you can say the test tube in which we have the most concentration of copper sulfate, or the 1%, will have the highest concentration of hydrogen peroxide. Why? Because the enzyme activity will be the least, because copper sulfate is the it is the inhibitor. Okay, so when you add a drop of P is added to hydrogen peroxide solution, you will see a pink color that quickly turns colorless, and P reacts with the hydrogen peroxide. So you will continue to adding P one drop at a time until the end point is reached. And what is the end point? The end point is when the pink color stays at least for 5 seconds. Okay, so now it is possible that the pink color will, and actually it will remain after 5 seconds. Okay, but you only have to wait for 5 seconds. Okay, not more than that, because it will disappear and it will actually add obscurity to your results. So you will count the number of drops to reach the end point. Now also note this point. Okay, this is, you can say, error of the experiment, because adding drop by drop is a difficult task, because the hands are also a little bit shaky. Also, some of the needles or the syringes are very hard, and when you try to add a drop, so more than one drop can be added. Okay, now the examiner is telling you that the greater the concentration of hydrogen peroxide, the more drops of P are needed to reach the end point. So now you should have this in your mind, that if more drops of P mean that there is more hydrogen peroxide, this means that 1% or the highest concentration of copper sulfate will have, actually it will be needing most drops of P. Why? Because it has the highest amount of hydrogen peroxide, because the enzyme activity was the least. Okay, so less substrate would have been converted to water and oxygen. Now he is telling you that carry out steps 8 to 14. Fill the syringe labeled P with solution P. Wipe outside the syringe with a paper towel. Hold the syringe labeled P over the test tube containing the lowest concentration. But you have to do this after you actually do the whole experiment. Okay, you actually you just have added the sulfuric acid, you just have finished this one. Okay, then you have to do it with all of the syringes, all of the test tubes. Okay, so you have to add the P to each of the test tube containing the lowest concentration. Now why are we starting with the lowest concentration? Because it will be needing the lowest drops. Okay, and actually it will waste less time. Okay, but you do not need to know this. You will not be actually knowing in the experiment, and that it will actually, which one will have will be needing the most drops, or the lowest amount of drops. Okay, but just because I know this from before, I'm just telling you so that how you should have the idea in the experiment. Okay, so you will be starting off with the test tube with the lowest concentration of copper sulfate. So this means that you will be starting off with 0.0625% and release one drop at a time, okay, of P into test tube. Shake the test tube to mix. Observe the color to see if endpoint is reached. The endpoint is when pink color stays for at least 5 seconds. Repeat steps 10 to 11. Count the total number of drops released until the endpoint is reached. You may need to refill the syringe with P. Record the number of drops of P needed if the endpoint has not been reached. Within 30 drops, record the result as more than 30. Okay, so repeat steps 8 to 13 with each of the other concentrations of copper sulfate solution prepared in step 1. Okay, so I hope you have just understood till now. Now we have to record the results in the table. Okay, so for the table, you should first of all let me just make a table. Okay, so for the table, you should make the table with the pencil, and it should be the points, the end points, or you can say these corners of your table should be closed. Okay, and also it should be drawn with a ruler. It should not be you can say it should be straight. Okay, it should not be something going like a line going like this and then this. Okay, it should be straight. Then you have to make 2 columns, one for the different concentration of the copper sulfate, and also for the drops that are needed. Okay, because they have told you that you have to record it in the A part 2. So I will make a line in between to just make 2 columns, and then one of the row will be for the headings, and then we have 5 concentrations, so I will have 5 rows. This will be 1, then the second, then the third, then the fourth, and the last one will be the 5th. So I will just reduce the size of this table. Okay, so this will be perfect. So something about this point. Okay, so now let's just first of all make the headings. So what will be the headings? The first heading will be concentration of copper sulfate solution. I'm just writing the formula because I don't have enough space, but in your exam you will write the full form. Okay, not any formulas, because this is not the chemistry paper, it is the biology paper. So concentration of copper sulfate solution, okay, which is C, and you also have to mention the units. Okay, you also have to mention the units. Let me just select this. You also have to mention the units, which are percentage sign. Okay, and then the second one will be the number. Let me just write number. So the number of drops of P needed to reach end point. Okay, so this will be your heading for this one. Okay, now how we start with the concentration. So in the first one, you will actually be writing the concentration, and your concentration should always be in ascending order. Okay, so what do I mean by ascending order? So as you go down, it will increase. Okay, it will increase as you go down the table. So we will be starting with the lowest, so this means 0.0625. Now most of the students do the mistake of adding the percentage sign here, which is totally invalid because you have already mentioned the unit in the heading. Okay, so you just have to mention the concentration. So it will be 0.0625, and then 0.125, then 0.25, and 0.5, and then 1%. Now you have to actually write the number of drops. Okay, so what I told you is that the more the concentration of copper sulfate, so it is the inhibitor. This means that more is the hydrogen peroxide concentration, which actually means that more drops of P, or you can say KMnO4, which is the potassium permanganate, are needed. So for 1% I will just write the highest amount of drops. Let's just pick 27, and your drops should be in whole number, okay, not in, you can say, decimal. It should be in whole number, otherwise it will be discarded. Then we will have 23 for this one. Let's just pick 16 for this, about 10 for this, and 6 for this one. Okay, so I just picked up some random values. Then they are telling us that describe the effect of changing the concentration of copper sulfate solution on the concentration of hydrogen peroxide remaining in the test tube. So you will know all of this just by doing the experiment. Okay, just because I know this from before, I have just picked up some random values. Okay, so concentration of copper sulfate solution on the effect of it on the hydrogen peroxide solution. So how we are going to be reducing? Now you can see in the table that as the concentration of the copper sulfate solution is increasing, the copper sulfate concentration, as it is increasing, the drops of KMnO4 is increasing. Okay, and it means that the hydrogen peroxide concentration is also increasing. So what I will write is that increasing the copper sulfate solution increases the concentration of hydrogen peroxide remaining in test tube. Okay, and by now, by this question, you should have actually figured out the trend in your experiment. Okay, so just keep an eye on that. So increasing the copper sulfate solution concentration increases your concentration of hydrogen peroxide remaining in the test tube. Now describe one source of error in step 10 to 12. Now remember, I actually marked one of them, which was, I think I just this is what I marked. Okay, so from 10 to 12. Okay, so from 10 to 12, just read it again. So hold a syringe labeled P over the table test tube containing the lowest concentration, and then release one drop. Okay, now you can also just mention this one, that it is difficult to release one drop at a time. And you can also write that the size of the drop varies. Okay, one can be larger, one can be smaller, and you can also write about the end point. Okay, and you can also write about this end point, that it is difficult to observe when the end point is reached. Okay, so you just have to stick with one of them. So I'll be writing about the size of the drop. So I will write difficult to release one drop at a time, as size of the drop varies. And the second one, actually I'm just writing two of them so that you should just keep them in your mind. Okay, so difficult to observe when the endpoint is reached. And in most of the papers, they ask you about the improvement, so you can just write it that you can use a colorimeter to reduce the endpoint. And for the drops, what you can do, that instead of measuring the drops, you can use you should actually measure the volume. Okay, so that can be the possible improvement, but in this paper I don't think so they have asked. Okay, so now they are telling you that river water contains sometimes can be contaminated with the copper sulfate sulfate from the factories. You will use the same procedure actually, and you will estimate the concentration. And how you will estimate? Actually, you will do the same procedure. You will actually count the number of drops for this one, and then you will compare with that table. Okay, so that between which of the concentration is the number of drops lying. So you have provided with the materials. Okay, so this will be the beaker that will be labeled with R, and then you have to do the same experiment with this one. You have to put 1 centimeter of cube of R into test tube, and then you have to repeat the same steps. Okay, first of all you will add the yeast suspension, then you have to the copper sulfate is already present in this solution, and then you will actually add the let's just take you to the steps 5 to 12. Then you have to add the hydrogen peroxide solution, and then you have to wait for 5 minutes, and then the sulfuric acid, and then you have to count the number of drops. Okay, so let's just suppose now they're saying that set a number of drops needed to reach the endpoint. So let's just suppose that we have 4 drops. Okay, so now what you have to do, you have to compare that where are we actually having 4 drops. Okay, so 4 drops will be lower than this. Okay, so if we are actually saying that 4 drops, so it means that concentration lower than copper sulfate solution, and in the case you actually got about 13 drops. Okay, so it will be between this. So you just have to compare it according to your experiment. Okay, so now if we just assume that 4 drops, so it will be less than 0.0625% concentration. So use your result to estimate the concentration of copper sulfate in the sample of river water. So let's just take it less than 0.0625, so it will be 0.05, and then the percentage sign is mandatory. Okay, otherwise you will lose the mark. So I hope you understand this. Let's move on. So now the B part says some scientists investigated a possible treatment for controlling the blood sugar levels in the humans. The scientists measured the effect of inhibitor found in the green tea on the activity of the enzyme sucrase, the enzyme hydrolysis the sucrose into glucose and fructose. So now you just have to make this graph. Okay, so I've already made a video on different type of graphs. Okay, how you have to identify the dependent and the independent variable. The one on the left hand side is mostly the independent variable. Okay, this is the independent variable which is going to be plotted on the x-axis, and this is the dependent variable which is being dependent on the independent, or you can say the concentration of inhibitor. So we just have to first of all choose the scale. Okay, so what is the range on the x-axis? The highest one is 0.5, and the lowest is 0.5, and the highest is 2.5. So let's see how many big boxes we have. So the maximum distance that you can take between two calibrations is 2 boxes. Okay, this is the maximum. If you take more than this, your scale will be discarded and you will lose your marks. Okay, you can either take 1 box, which is actually 5 small boxes, or you can take 2 boxes. Okay, so for this time I am taking 2 boxes. So I have 1, 2, 3, 4 and 5 big boxes. So it will be, let's just it will be equally divided. 2.5 divided by 5, so it will be 0.5. So this will be 0.5, then 1.0, 1.5, then 2.0, and this will be 2.5. And you also have to mention the axis full. Okay, with the units. So concentration of inhibitor, milligram per centimeter cube. So this will be concentration of inhibitor, milligram per centimeter cube. And then for the first one, the US value is 8 and the highest is 52.5. Let's see how many boxes we have on the y-axis. So it is 1, 2, 3, and actually one big box is 10 small boxes, or you can say 10 small units. Okay, then 4, and then 5, and then 5.5. So it actually means 55 units. So if you divide 52.5 by 5.5, it will be, you can just equally divide it by 10. Okay, so it will be 10, 20, 30, 40, and this will be 50, and this will be actually 55 one. This is the 55, but we actually don't write it because we actually are taking the calibrations of with a distance of 10. Okay, so 52.5 will be something about this point. Now always and always read the second question. So draw two lines, okay, because most of the time they just try write up to draw you can say a curve, okay. So then what you can do, you can actually just connect the lines with the straight lines, or you actually make a curve like this, okay. So you can do either of them, but actually it is less common. But now you can see that he is asking to draw two lines on your graph to show the concentration of the inhibitor that cause 24% inhibition of sucrase, okay. And actually when you are drawing two lines, it actually means that you have to draw two lines of best fit and you have to intersect them, okay. So so let's see. First of all, we will draw them, okay. So first of all, we will make over graph, and then we will be drawing these two lines to show the concentration, okay. So it will be, first of all, we have 0.5% on the x-axis and 8 on the y. So 8 will be something about this point, and 0.5 is this point. You just encircle this, or either you make a cross, small cross, okay, like this, okay. So you can just do it in any way. Then 1% and 27.5. So 27.5 and 1%. So 27.5 is something about little less than 28 and between 27. So this is the point, okay, and just circle this. Then we have 1.5 and 44.5. So 1.5 is this line, and then 44.5 will be something about this point, okay. Then we have 2 and 51. So 2 and 51. So 51 is here, and 2. So this is the point, and then 52.5 and 2.5. So 2.5 is the last line, and this is 52.5, okay. Now just join them with straight line, or either you can just make a curve, okay. So this will be the straight lines. You just have to join the points with the straight lines, okay. So I hope that you already know this. In the case if you do not, you can just watch the whole playlist on the practical, where I have shown the type of graphs, okay, and how you have to make the graphs and the tables and the drawings for the plan diagrams, okay. So this is how you join, okay, and then you will be drawing the two lines to show the inhibition at 24% of inhibition of sucrase, okay. So concentration of the inhibitor at 24%. So 24 will actually be, let's just draw it with the other color. So it will be this, okay. This is 24, and just actually we draw it with the dotted line to show this the point. This is the point, okay, and then just trace it down to the x-axis. This is the point. So it will be let's see, so 0.5 divided by 10. So we are actually having one small box equal to 0.05. So it will be 0.05 into 2 and minus 1. So 1 minus 0.05 into 2. So it will be 0.9 actually, okay. This will be 0.9% concentration of inhibitor. So you just have to show this on the graph, okay. Then they are telling you suggest how the inhibitor in the green T reduces the activity of the enzyme sucrase, okay. Now you can actually just use the concept of the non-competitive inhibitor, or you can just write a general statement that how it actually stops it from binding to the, so it actually stops the substrate from binding to the active site of the enzyme. It stops the formation of the enzyme substrate complexes and reduces the activity of the enzyme, okay. So or either you can also write that it binds to the allosteric site of the enzyme, it distorts the shape, and it can no longer bind to the substrate, and as a result less enzyme substrate complexes are made. Okay, so I will be writing that. So you can say the inhibitor in the green tea binds to allosteric site on sucrase distorting the shape of active site, so it can no longer bind to substrate, decreasing the formation of enzyme substrate complexes, and hence reducing the activity of enzyme. Okay, so this will be your answer. Then the last part of this question says the scientists calculated the percentage inhibition of the sucrase by measuring the concentration of reducing sugars in the solution after 5 minutes. Describe how the scientists could determine the concentration of reducing sugars in the solution. Okay, so now this type of question, what you have to do, you just have to give a scheme of work, or you can say just a plan investigation of how you are going to be actually reducing the concentration of a solution, of a reducing sugar solution, in the same way, actually in the way actually that we did. Okay, so now what is the test for the reducing sugars? It is the Benedict test. Okay, it is the Benedict's test. Okay, and now what you will be doing, actually you will prepare different concentration of the reducing sugars, okay, of the known concentration. For example, we will do the dilution, we'll prepare 1%, 0.8%. You can either do seal dilution, you can either or you can do the simple dilution. So it will be 1%, 0.8%, 0.6, 0.4 and 0.2. Then you will actually time them. Actually what you actually do in the Benedict test, you can also mention the Benedict a small you can say section of the Benedict test, that you will time each of the concentration, and then you will do the same with this unknown concentration of the solution of the reducing sugars, and then you will compare its time with the time of the known concentration to reduce its concentration. Okay, so I'll be writing the same thing. So prepare 5 different known concentration of reducing sugars, which is 1%. Okay, you can just give the value 0.8% and 0.6%, 0.4% and 0.2% using serial dilution. Okay, serial, not seed, simple, using serial dilution, because it is more effective when you are actually want precision in the concentration of a sample. Using serial dilution and perform Benedict test and note the time taken for each concentration to reach endpoint. Repeat the same for solution of unknown concentration and compare its time with known concentration to deduce with known solutions actually to deduce its concentration. Okay, so this will be your answer. Okay, so the first question is finished. Now we have the second question, which is M1 is a slide of a strain transfer session through a leaf. Now they are given you the leaf. Okay, this is the recent paper, and I am also expecting the same thing for the October-November. Might be possible that they give you an experiment on the osmosis, but most probably they are going to be testing the same for the same year. Okay, so draw a large plant diagram of the region of the leaf in the M1. So indicated by the shaded area in figure 2.1, and then use one label line to identify the lower epidermis. Okay, so now you can just, I have just picked up some slides, okay, and I have drawn the plant diagram of this. So I actually have just forgotten that which of the slide I used to make this diagram. Okay, so I've just given these images for reference. Okay, so you can just use any of them. Okay, let's just shift them towards the paper. Let's make this a little bit large, and this will be, let's just adjust this. Okay, so now this is our plant diagram, and we will use the ruled label line. Okay, always use ruled line, and that is just touching the structure that you want to let me just erase this. I just made it by mistake. Okay, so this will be your ruled label line, and just adjust it so that it just touches the structure that you want to label. Okay, in most of the cases you also have a cuticle on the upper epidermis, but in some cases you also have it on the lower epidermis. Okay, so this will be your lower epidermis. Okay, so just this. Let's move on to second part, which says observe the xylem vessel elements in the leaf of the M1. Select a line of 4 adjacent xylem vessels. So I have already just made a full video on the drawings of the cells of the xylem wells of the you can say the mitotic cells in the root hair cells, also for the for the bronchus, and for the different structures for the dicot stem and for the root. Okay, so you can just watch that video. I have also covered the xylem vessel elements in that one. Okay, so for this one we'll now be doing the calculation. Okay, so now they've given you the figure 2.2 in the part in which you mostly compare it with the first one. Okay, so figure 2.2 shows a photomicrograph of transverse section through a different leaf from that on the M1. Okay, so now they have given you this image, and then you have to identify 3 observable difference other than color between the section on M1 and section in figure 2.2. Now you can see that over plant diagram this actually have you can just see that it has the stomata. Okay, it has the stomata, while this one you cannot observe any stomata. Okay, so the first feature that we can observe is that no stomata are present. Okay, then the second one, you can just see these protruding structures. These are actually your trichomes. Okay, and these are actually present here but not in the slide of M1. Then the third thing is the shape of this leaf. This leaf is actually curled okay, and that one is not curled, okay, that is just straight. And the third thing that I will say, and that is not mostly covered by the teachers, is that the width or the area that is being covered, the width covered by mesophyll tissue, okay, the width covered by mesophyll tissue. So this is also a valid point. You can see that the area here is less and it is more in the slide of M1. Then the third thing that you can see here is that the vascular bundle, okay. So here the size of it, the size of the vascular bundle is smaller here, while on the slide on the M1 we have a larger vascular bundle, okay, and in the most of the cases you have numerous amount of vascular bundles, okay, but here we only have one, okay. You can also write it about the number. So I have given you more than 4 observable features. You can write any 3 of them, okay. And the part C says that figure 2.3 is the same photomicrograph as that is shown in the figure 2.2. Now use the scale bar on figure 2.3 and the line DE to calculate the actual length of the gap between the ends of the leaf. Show your working, include units, and give your answer in micrometers. Now these type of questions are not more often, because they just tell you the magnification, then you just measure the line and then use the formula to find the actual length, okay. But here what they have done is that they have given you the scale bar. Now what is the scale bar for? Now the scale bar tells you that this is the, now they have given you this length, okay, 145 micrometer. So this is the actual length, but of what? This is the actual length of this line, okay, and this scale bar is used to find the magnification, okay. This is used to find the magnification. So this is the, now they are telling you that the actual length of this line is 145 micrometer. Now you just have to measure this. So the length of this is 2.9 centimeter according to me, and you just convert it to micrometer, so it will be 29,000 micrometer, okay. It will be 29,000 micrometer. So you will use the formula for magnification made over actual. So 29,000 divided by 145, so it will give you the magnification. So the scale bar is always used to find the magnification, okay, so remember that. So it will be 200x, and then you will actually use this magnification to find the actual length of this line DE. So I just measured this is 8.3 centimeter. If you convert this to micrometer, it will be actually 83 millimeter. So if you convert to micrometer, it will be 83,000 micrometer, okay. This is the measured length, and then you have to find the actual. So magnification is equals to measured divided by actual. You just rearrange the formula, it becomes actual is equals to measured divided by magnification. So it becomes 83,000 divided by the magnification, which is 200. This will be 415 micrometer will be your answer for this part, okay. So this is how you do. That is why he has given you 4 marks, because most of the students do not know how to actually use this scale bar, okay. They think that this is the actual length of the line, but you just have to use it for the magnification. They are just giving you an aspect ratio of the real to the one in the actual. So this was all for today. We just have completed the whole paper, okay. So I hope you like this video. If this was beneficial, do share with your friends, and also do subscribe to the channel and like the video. I'll be meeting you in another video, and wishing you a good luck for your paper on the 7th of October. Signing off, this is Hasvora Education. I'll be meeting you in another video. Till then, take care and goodbye.